فيس كونتراست تكنيك كونتراست.
الدكتور احمد عبد الواحد هيقدم presentation بعنوان Functional MRI Overview.
Good afternoon. My dear professors, my colleagues, I'm deeply honored to be amongst you today. It's a quite complex subject, so let me guide you through this very brief journey of the fMRI. So at the start, what is fMRI? It's a class of imaging techniques to demonstrate the regional and time-varying change in the brain metabolism. These metabolic changes can be consequent to a task-induced cognitive state change, or as a result of underregulation or upregulation process in the resting brain. So what are the indications? Of course, most important thing is pre-surgical planning for intractable temporal lobe epilepsy or brain tumors. It's gaining popularity in the cognitive neuroscience and the clinical psychiatry nowadays. So fMRI is of course based on MRI, nuclear magnetic resonance imaging, coupled with gradient magnetic field. This will create image that can incorporate many different types of image based on T1, T2 flow, or of course susceptibility images. So in order to understand the particular contrast mechanism predominantly used in fMRI, we have to understand the brain metabolism. All the process of neuronal signaling, starting from the action potential generation till the postsynaptic release of neurotransmitter, will require energy using ATP. This nucleotide produced by the mitochondria resulting from glycolytic oxygenation of the glucose with production of carbon dioxide as a byproduct. A very simple maneuver when the region of the brain is upregulated or activated by any cognitive task such as finger tapping or smelling a perfume or hearing a very loud voice. The additional neuronal firing and other increasing signaling process will result in local increase in the energy demands. This requirement in turn resulting in upregulated cerebral metabolic rate of the oxygen in the affected brain regions. Simply, the local stores of oxygen in the tissues adjacent to the capillaries are transiently consumed by the glycolysis and the waste products will accumulate. These waste products will induce a vasomotor reaction in the capillaries and arterioles that will result in dilatation of the vessels. Subsequently, there will be increased blood flow to restore the local O2 deficit. So we have a transient local O2 deficit. With a vasomotor reaction, it will be increased blood flow to restore this O2 deficit. However, for unknown reason, the oxygen is delivered more than needed for the offset of the increase in the CMRO2. As a result of the neuronal upregulation, it will result initially in the buildup of the deoxygenated hemoglobin and decrease in the oxyhemoglobin in the intra and extravascular spaces. And then a few seconds later, there will be the reverse. The vasodilatation will result in increase in the oxyhemoglobin and decrease in the deoxyhemoglobin. This is called the hemodynamic response to a neuronal event. So when there is an increased neural activity, there will be increase in the local blood flow, the cerebral blood flow, and there will be changes in the oxygenation concentration, what we call the blood oxygen level dependent or the pooled signal. Both of these changes can be detected by the MRI. This change in the cerebral blood flow can be observed using injected contrast material or non-invasively using the arterial spin labeling. However, the arterial spin labeling will suffer from the reduced sensitivity, increased acquisition time, or increased sensitivity to motion compared with the pooled signal. For the pooled signal, when the fully oxyhemoglobin is a diamagnetic substance, so it's magnetically similar to the brain tissue. However, the fully deoxyhemoglobin is a very paramagnetic substance. This paramagnetism will result in a local, very small gradients within the magnetic field. This gradient will cause loss of signal or drop of signal in the T2 and T2 star sequences. Of course, we use a gradient refocusing echo, which will make it sensitive to T2 star and T2 effect. Most of the time, we use 1.5 Tesla and 3 Tesla using T2 star contrast as predominant and in the largest individuals, while the high field strengths the diffusion weighted contrast of T2 relaxation becomes more important because signals are generated preferentially in the capillaries and in the tissues with a spin echo acquisition. Since most fMRI is currently performed at 3 Tesla machines, the pooled fMRI utilize the gradient echo methods more than the spin echo methods because of time, of course, and will be very affected by the T2 star effect. Right. What we do then? We seek to induce a different neuronal states in the brain, like visual, auditory, or any other stimuli. And this is manipulated during the scan. We generate then activation maps obtained by comparing the signal recording during different states. Therefore, it's important to collect each image in a snapshot mode to avoid the head motion, the physiological process of respiration, cardiac motion, in order not to inject a noise signal unrelated to the process. Generally, most fMRI is performed using the echoplanar imaging, methods which can collect data for two-dimensional image approximately in 60 milliseconds. The typical fMRI task activation experiment utilizes visual, auditory, or other stimuli to alternately induce 2 or more different cognitive states in the subject, while collecting the MR volume continuously is described. With 2 condition designs, one state is called experimental condition, and the other state is called control condition. And the goal is to test that there is a difference between the 2 states. Using block design, the trials are arranged to alternate between the 2 experimental and the control conditions, with each block typically taking a few seconds. Here we can see that we have 2 stimuli, A and B, and the acquisition is made continuously and rapidly using this pooled signal. And then we have 2 images, the A state and the B state images, and then the difference between the 2 images is computed to perform the contrast map. And then this contrast map, using computer software, is analyzed to produce activation map where it shows the area of firing responsible for this maneuver, like smelling or snapping the finger or what else. The block design is optimum for detecting activation, but there's another design called the ER design is superior when characterization of the magnitude or timing of the hemodynamic response is desired. In this ER design, the task events are relatively brief. At non-contrast inter-trial intervals with longer periods of control condition, which allow the hemodynamic response to return to the baseline rapidly. In some studies, this is very straightforward, such as the use of sensory task for a pre-surgical mapping, where the goal is only to localize the activation that is important for the brain function that can be maintained after surgery. Let's skip this analysis. What are the strengths of fMRI? The primary strength of fMRI is relatively high spatial resolution. Also, it is very non-invasive, and it provides high resolution of the anatomic scan in the same session to use for localization, vessel identification, development of maps of white matter connectivity through the use of diffusion tensor imaging. But what about the weakness of fMRI? Because bold contrast or bold signal derives from the sluggish hemodynamic response to neuronal activity, the metabolic changes will be significant weakness in its low temporal resolution. Another problem is the signal dropout or the spatial distortion in the frontal, orbital, and lateral parietal region. This is because the interface between the air and the brain tissue will result in 9 parts per million difference in the magnetic susceptibility. Another weakness is the scanner loud noise associated with switched magnetic fields, which can cause confounds in the studies of audition. The high magnetic fields require customized stimulus, delivery, and subject response system, again limiting the flexibility and complicating multimodal experiments such as the concurrent EEG recording at time of scan. What are the futures of fMRI? While the modest research effort will continue improving acquisition technology, the bulk of research and development of fMRI has shifted to its application to answering more complex questions in the cognitive neuroscience. One promising area is that of using activation maps as input to classify and state changes algorithm to predict and classify cognitive behavior of the humans, such as predicting the violence behavior in criminals. The emerging use of fMRI include the development of quantitative measures as a biomarker for diseases. Finally, the feedback from real-time fMRI has been shown to allow subjects to learn pain reduction strategies, enhance sensory motor control, and to control relevant brain regions in a mood disorders experiment. Thank you.
بنشكر الدكتور أحمد على